It travels through our body's respiratory tract. In this case, the coronavirus travels to the alveoli. The spike proteins of the coronavirus bind to the ACE2 present on the host cell plasma membrane. Once bound, the ACE2 facilitates receptor-mediated endocytosis. As a result of this process, the virus is pulled into the host cell. Once inside, uncoating happens, and all the proteins are removed, leaving just the genomic RNA sense strand in the cytoplasm. During translation, using the host cell machinery and using open reading frame 1A, polyprotein 1A is made. As translation is occurring in the middle of open reading frame 1A, a frame shift can occur to open reading frame 1B. That's how the polyprotein 1AB is produced. Polyproteins 1A and 1AB are broken into smaller proteins. These proteins form the replication transcriptase complex and combine with the genomic RNA sense strand to facilitate replication.
Replication results in antisense RNA. This RNA can be used for two things. The first process is replication to create sense strand RNA. This RNA will be packaged into the virion progeny. The second process is transcription, to produce multiple mRNAs that code for the structural proteins of the coronavirus. Using this continuous transcription, these multiple RNAs of various lengths are produced.
RNA polymerase, an enzyme produced by the sense genomic RNA to transcribe this antisense RNA binds
the genomic antisense RNA. There is a leader sequence called the TRS. This is followed by the region that codes for the replicase proteins, and lastly, the body TRS. Transcription for the structural proteins mRNA starts at 3', goes to the corresponding body TRS, jumps to the leader TRS to finish transcription. Everything between the body TRS and the leader TRS is emitted from the mRNA. This is why it's called discontinuous transcription.
This results in multiple mRNAs of different lengths. All of these various mRNAs are translated in the rough ER into viral proteins. The viral proteins, along with the genomic RNA, are sent to the Golgi apparatus, where they are put into vesicles and packaged as the virion progeny. Then by exocytosis, the progeny is expelled out of the cell. This fully mature coronavirus goes to infect other cells in the body.